hello everyone. My name is Sandra and I'm a grad student at Loyola University Chicago. And today I will be taking you on a journey about the diversity of Staphylococcus epidermidis in the female microbiome. So to first get everybody oriented, I wanted to introduce my favorite microbe, and hopefully after today, everybody's favorite microbe, Staphylococcus epidermidis, or Staph epi as I like to call it. So it is a common microbe that is found in the skin microbiota. Furthermore, it is considered the good guy. It's a commensal microbe that helps evade opportunistic and or pathogenic bacteria. Yet, if given the chance, it can act as an opportunistic pathogen and cause infections such as staph. To begin this origin story, we're going to ask ourselves, are S. epidermidis strains created equal? We know or we think that they might have arisen from a single ancestor. Whether you were born and or extracted from your mother's womb, you get strains from the people that you're around. We see that neonates' microbiota can closely resemble their mothers, the same strains or even identical strains. Then you get into the more environmental strains, such as paternal figures, pets, and other environmental aspects. So, are all S. epidermidis strains created equally? We know that there is diversity amongst strains. However, most of the genomes that we know that are publicly available are truly biased to the skin sampling. My colleague took down all the publicly available S. epidermidis genomes recently, and here at the bottom to orient you all are the isolation sources on the x-axis, and on the y we have the counts. You see that there is a disproportionate amount of skin sampling or S. epidermidis that is found in skin isolation sources compared to any other type of anatomical site. Why might that be? We might have a preconceived notion that S. epidermidis is only consistently found on the skin and nowhere else, which is not true. This leads me to my question. Do S. epidermidis strains from the skin resemble strains elsewhere? As most scientists do, you do what you know. My lab focuses on the urogenital tract or the urinary tract. These samples were collected from our colleagues at the Loyola Maywood campus. They were sent over to our lab at Lakeshore, where I extracted them and sent them off for whole genome sequencing. Here to orient you is a phylogenetic tree, which was composed of the pangenome of the samples that we got after sequencing. And once the pangenome was made, we looked at all of the cores or all of the conserved genes from these samples, which is what is constructed in this phylogenetic tree. The most interesting aspects of this phylogenetic tree are that 1 and 9 are samples that come from the same participant. And you have an ANI value, or an average nucleotide identity value, of 99.99. In the microbial world, you could say that these are very genetically similar, if not identical strains. However, you have samples 3, 7, and 10 that have only a 99.32% ANI. So although they are from the same isolation source, they are from the 3 different participants that we got. What does this mean? What is the bigger picture? Although this is on a smaller scale and we only have an N sample size of 7, we would assume, or we did in this case, that samples found in the same participant or patient are going to be more closely linked than amongst isolation sources between different individuals. This is where my study comes in. My study of 2 years focused on the genomic and phenotypic differences of S. epidermidis from different sites. The sites that I decided to focus on was the nasal, the skin, the urinary, and the oral microbiomes. The reason for this is we saw that the urogenital tract, which is more closely related, has similarity in strains. How do those fare in distinct anatomical sites that are, first of all, different based off of distance, but also off of things as pH, temperature, salinity, and openness to aerobic or anaerobic conditions? The requirement to be in the study was to be a healthy 18 to 25-year-old female at the Loyola Lakeshore campus and to have not taken antibiotics in the last 6 months and clearly present without any type of illness at the point of collection. The point of collection would include a participant coming in and seeing my smiling face as I gave them a urinary cup. Once they came back with that urinary cup, they self-swabbed in 3 other distinct places. The oral was the inside of the cheek, the nasal inside of the nose, and the skin was a forehead sample. I will now show you the distribution of these samples. So bear with me as I go through it all. This is a heat map with a presence or absence type of data. At the bottom, on the x-axis, we have all of the participant samples from 100 to 200 because I personally like that range when I take samples. And then on the y-axis, you have all the different isolation sites. Here, you might see a distinction between the oral microbiome, that there is a lot of S. epidermidis that is not present. This may, first of all, be due because there is no S. epidermidis present in the oral microbiome, or simply we may not be extracting correctly. Another thing to note is that the skin microbiome, as you would assume, should be readily extracting S. epidermidis, but that is not the case. Furthermore, we also see that most participants have S. epidermidis in more than one anatomical site, meaning that S. epidermidis can be found in almost any type of anatomical niche, not only specific to the skin. In order to further evaluate these strains, we decided to look at a pangenome. A pangenome is the entire set of genes from a specific species, and this species in this case would be an S. epidermidis. This is my full pangenome of the around 80 samples that I've shown you before that were nicely distributed in a heat map. Although on the left, it may be a little bit convoluted, the black dictates or indicates the core that is conserved amongst all of the samples, while the white and or clear sections are the accessory genomes or things that are not actively conserved amongst all of these samples. It is really hard to see anything of meaning until you make it into a phylogenetic tree. On the right-hand side, you have the phylogenetic tree with the inside ring being samples that we have collected and the outer ring indicating isolation sources. The key things to know in this presentation or from this phylogenetic tree is that we have the same assumptions or things that we saw from the preliminary study. We do have isolation sources that cluster more together, and we also have samples from the same individual that cluster together as well. The one thing that we can conclude is that there is no clear clustering based on one or the other, solely from the core genome. This led us to assess things like an enrichment matrix. Here we have COGs, or we have clusters of orthologous genes. Here we see that all of the isolation sources have some kind of significant COGs prevalent singularly in one anatomical site compared to the others, except for oral. There were none that were statistically significant, giving a p-value of less than 0.05, which was very interesting. So next steps of this whole study is to look at the functionality of these genes as they're related to singular isolation sources and see if they can be found in transient strains across the body. Furthermore, look at microbial populations between different anatomical sites, meaning not only looking at S. epidermidis strains, but more of the populations that make up these diverse microbiomes. Throwback to the oral microbiome, we saw that there was no S. epidermidis strains. With investigation of microbial populations through 16S or RNA, we can check if S. epidermidis predominates in the oral cavity in some aspect or is even found, or potentially not. With that, I wanted to acknowledge my lab group, the Patanti lab group, the participants who were so nice in donating their samples and time, and Loyola University Chicago for funding my research. Thank you.
Great. We have plenty of time for questions. Dan?
Thank you. I always like these studies. Can Can you go back to your tree?
Yes. Yeah. This one?
So this was a tree that you built using an alignment of cork.
Yes.
Did you try doing a presence absence? In other words, you could, using the
Mm-hmm.
gray hat, you could say, you could make a vector that says this gene is present here and absent there.
Oh, that is a
And
very
use
good
that as well.
suggestion. Yeah, we have not, but I will take that into account.
You're seeing some things like,
Mhm.
but that are maybe, I mean, artifacts is too strong, but that's because you're only sort of measuring relationships on some axes, and there are other ways in which you can measure them as well.
Thank you. That is so insightful. Yes.
I had a question about the cogs, because I
Mhm.
don't really know a lot about what that actually means. Is that indicating that there is like niche specification within S. epidermidis?
Yeah. So it's yes, but it's more in these distinct isolation like sites. So it's nutrients, cell cycling, things that upkeep the strains in the specific niche that they are found in.
Those are the genes that are
Served solely in that specific anatomical site. That is like statistically significant. Yes.
So any other questions? I guess I have one.
Yeah.
On the tree on the previous slide,
Mhm.
do you happen to know what the significance of that really far out root is?
No.
You don't know.
That is under investigation. That is a very valid
That
question.
was the first thing I saw. I'm like,
Right.
yeah, okay.
Great.
So,
Mm-hmm.
do you have isolation source on the, can we tell from this particular plot on the left which isolation source in particular?
Mm-mm.
I'm looking forward to your next chapter.
Thank you. Thank you. Perfect.
Great.
Great.
Any other questions? Oh, we have one in the back.
Yes.
I noticed that you have a lot of subjects who had got bacteria in the nasal cavity, but not in the oral cavity.
Mm-hmm.
So just wondering if any environment like in the oral cavity, any big chemical factor that might have inhibited the existence of the bacteria in the oral cavity.
That's an excellent question. It has come up. So the question asked was, we see S. epidermidis strains in the nasal, but not the oral cavity. Are there any type of chemical or biochemical aspects of that anatomical niche that make S. epidermidis not able to grow or be collected? That's a question. Perfect. We have thought about this before, and that might just be that there isn't any, or that the way that we are collecting is just not translational for S. epidermidis from the oral cavity to grow. However, I do not know about the more biological aspects because all of these anatomical sites are diverse in multiple ways, yet you see growth from urine, nasal, and skin. Mm-hmm. Yeah, no, that's a great point.